today we'll have two speakers on this ongoing seminar series. Ute Hentschel Humeida and Luisa Figueiredo, that I'm very thankful for accepting this invitation. So we start with Ute, that is in Germany, and Ute did her PhD in the Scripps Institution of Oceanography, where she started to get interested in marine biology and work on nitrate respiration of hydrothermal vent worm. And then she moved for a postdoc in UC Santa Barbara, then a postdoc in the University of Würzburg, where she started a group, and in 2015 moved to GEOMAR, where she is now with the group. And since her postdoc, that she got interest in sponges and the microbiome of sponges, and which is a very interesting system to work on, because it's a very simple animal with a complex microbiota. And she has been contributing to the field, not only describing all of these complexity and communities, but also understanding functionally how do they interact by doing lab work, field work, and use of several omics techniques. And today she will talk about one of these projects on a phage protein that mediates interaction between the bacteria and the sponge host. And thank you, Ute, for coming to this talk.
All right. Thank you, Luis, for this kind introduction. So yes, so my talk is about sponges, bacteria, and phages, and what we view as a tripartite interaction. And as you just heard from Luis, my long-standing interest is in both symbiotic and pathogenic associations. And both facets will come to play in my presentation. So sponges, a few introductory words on sponges. These are animals. These are sessile filter feeder animals. That is probably the oldest metazoan phylum that we have. And we have fossils that date back to 600, 640. Some people even say 800 million years ago. And as you see in this image, these are filter feeders. This is a Caribbean sponge called Agulhas that pumps water through its surface and then to be exhaled in one of few ostruli. And the water that passes through the sponge is essentially sterile, while, as you may know, normal seawater contains about 10 to the 5, 10 to the 6 bacteria per ml, the size of your fingertip. So these are extremely efficient filter feeders. They are habitat forming, meaning that they give rise to communities of benthic critters, sea stars, sea urchins, small fishes. They are chemically dependent, and in fact, the history is that sponges have largely been studied by chemists who find them to be a very rich source of new compounds that may find applications in pharmacy or medical applications. People like my lab and many others are, however, interested in the rich symbiotic communities that are contained in such animals, much like we humans have huge amounts of microbes in our intestinal systems. And I also would like to point out that as of today, only a handful of sponge genomes have been published. And that's not very much for a phylum that may have 9,000, maybe 10,000 or more species. But we are currently working with funding from the Sanger Foundation and the Gordon and Betty Moore Foundation to generate up to 50 different sponge genomes, or I should say sponge holobiont genomes, because we are sequencing the microbial consortia as well. Now, when you take a closer look into a sponge, you see really not much. The animals are have no internal structure, no brains, no tissues, no bones, no nothing. It's really a three-dimensional matrix in which is embedded what we call an aquiferous system. It's basically the plumbing of the animal, where water is drawn through the surfaces and then percolates through these granulocyte chambers, and then to leave the animal, as we've just seen, in one or several exhalent chimneys. And again, 20,000 liters per day per kilogram are pumped through these animals on a regular basis. Now, when you take a closer look at the sponge, you see the interior, sometimes spicule cells that give structure to the animals. Like in the Caribbean, these animals can be as big as a wine barrel, maybe 1 meter in height. But what we also see are those cells here, basically phagocytotically active cells. I imagine they patrol through the interior space like Pac-Man, just kind of eating their way through and eating everything up that comes into their way. Now, a real picture from our model sponge Eclasina aeroproba is on the right side, and you see again the granulocyte chambers, what I call plumbing, where the water goes through, and then there is a physical barrier, and microorganisms, food particles, and the like have to pass through this barrier to enter the mesohyal tissue. I imagine as a gelatinous space in which we find lots and lots and lots of microorganisms, as you see here, and once in a while, but rather rarely, a sponge cell. So this would be one here, and this is one here. And for this reason, people like my lab would call sponges microbial fermenters, because in fact, up to 1/3 of the animal's biomass can be purely microbial. So these are really Eldorados for microbiologists. And when we got started some years ago, we basically set out to understand diversity in function and also biotechnological potential of such associations. And this is just a summary what my lab aims to do. We've been studying these enigmatic animals for some years now. We have an interest in studying mechanisms of interaction. It's largely about metabolism, but not always. We try to unravel ancient principles, where we try to understand or claim. We like to think that whatever we find in sponges may be relevant to higher organisms. And essentially, we try to understand how these associations function, evolve, and ultimately also influence moving ecosystems. And we always have an interest in these dynamics of symbiosis and dysbiosis, taking into consideration that our world is changing, temperatures are rising, and systems can, in some cases, move from a symbiotic to a dysbiotic state. So this is basically what we do. But today I want to tell you one story, and I have actually picked this one because I know that you guys as an audience are quite hardcore in molecular and cellular biology. So I want to tell you about our story on phages and phage proteins. And this story is really told by a former graduate student and now postdoc Martin Jahn, who's now in Oxford with Kevin Foster. And Martin was in my lab here in GEOMAR and also in Würzburg and published a really beautiful paper 2 years ago that even made the cover of Cell host-microbe. And I think this is the first time that a marine organism, for sure a sponge, has ever made any cover of any premier cell journal. So we are naturally proud of it. And what you see here for the is the granule, the mesohyal tissue, and in green the bacterial cells. And Martin, by 3D tomography, could even visualize certain cell compartments by 3D tomography of a poribacterium, that is a candidate phylum that we discovered some years ago that only exists in sponges. Okay, so that's, you can see I'm a little proud of this cover. But what is the research? So we are going from sponges to microbes to talk about phages and viruses. And these animals filter this massive amounts of seawater. And we learned by ways of metagenomics already in the years 2015-16 that remarkably the metagenomes that we obtain from sponges are enriched in defense mechanisms, restriction, modification, CRISPR-Cas, toxin, antitoxin, and the like. And first, we couldn't really make sense of that. Why should they be dependent? But then we started thinking and thought probably because as the animals filter, they bring in all this viral, free viral and phage DNA, and they constantly have to defend themselves against this mess of viruses. But the field at that time wasn't really ready, and in fact, only a handful of studies, largely by Nicole Webster and her people, have looked at viral diversity in sponges. Now, Martin then went to Barcelona with a collaborator, Lucia Peter, And they went diving and they picked 4 different sponge species in a nested sampling design, and also alongside seawater as a control. And then basically filtering seawater. I should say first, Martin separated the tissues, exterior versus interior part, and then purified viruses by centrifugation, DNA extraction, amplification, sequencing to produce 36 metagenome viromes for species and for replicates inside outside, totaling 142 gigabase of virome DNA. To our knowledge, this is the largest data set still to this date from marine sponges. So when we looked at the data, we found first and foremost a really very high degree of novelty in viral DNAs. And altogether, he assembled more than 4,000 viral contexts. We don't have names for them because viruses and phages don't have a marker like 16S or 18S. But he essentially had taken all our virome sequence data and mapped them against all publicly available viral databases that were there, Actinophage and whatnot. And what he found that much of what we got from sponges was entirely novel. So all marked in green are phage contexts from sponges mapped against black reference database. So in fact, only 3% of the sequence data could be annotated to a family status. And you will recognize some known viral families, such as the Phodoviridae, Myoviridae, Cephuviridae, marked in colors. But the vast majority, 97% of it, were unknown to all viral databases back in 2019. And considering that he had only picked up a handful or a dozen species, this is quite a remarkable finding. Well, we then went ahead and had a bit of a closer look. That is a dendrogram here, and we found second, and also to our surprise, that each individual sponge harbors its own virome signature. And what I mean here, that if you look at green, for example, these are the species Agellas, Chondrosia, that are seawater samples, and Apelsina. And what you can see here, that they not only cluster according to host species, but they also cluster per individual, because this is a mesohyl tissue and a pinacoderm. So that's the inside, the outside of individual 1, inside, outside of individual number 2, and so on and so forth. So again, I just told you a few minutes ago that sponges filter thousands of liters of seawater per day, and they don't really have an epidermal barrier or alike, and still they seem to be able of keeping individual specific viral signatures. To us, that was a surprise. We might have thought that since they filter water so massively that there would be a common signature, but we found a strong signal. And then we went one step ahead and basically asked, well, how can this be? What are the reasons? And we basically mapped the viral community diversity juxtaposed with the microbiome community diversity that we had obtained by 16S sequencing. And I think you agree that the majority of samples show that the virome maps exactly on the microbial community signature per sponge genus, and over large extent also per species. So I think what we can learn here is that these viromes are highly specific to the microbiomes from the corresponding sponge individuals. So it's basically mirroring the microbiome diversity. So then we went ahead and used in silico host prediction. There are tools out there that allow you to use CRISPR spacer or homology matches and allows you to map pages for their bacteria hosts. And in doing so, Martin could construct phage bacteria infection networks. And what we learned by this approach was that, yes, indeed, certain phages marked as red diamonds would indeed map, and they would be specific to certain bacteria that we knew as clear sponge symbionts. And such examples are chloroflexi, certain types of cyanobacteria, flavobacteria, but also the already mentioned poribacteria that have made it to the cover of Cell. So we have phages that are very specific to certain sponge bacteria, and alongside we have phages that map to what we know as bacterioplankton. So that is not that uncommon, I should say, because phages can be highly specific to bacteria, but this data showed us that the phage and viral diversity that we recovered was indeed specific to the sponge symbionts. Let me see. So I just want to point out here, we have written a subsequent paper that was then published in ISME, where Martin has constructed a new microscopy protocol, mixing correlative light electron microscopy, scanning electron microscopy, and FISH. And in doing so, he succeeded in basically finding the phages that are marked here in blue in serial sections back in the original sponge tissue context. So if you want to learn or curious about microscopy, I recommend to follow up on his paper, because the extent of specificity that you find individual phages in a sponge that is 10 kilograms is quite not trivial. But I want to continue with my story here. We talked about diversity, we talked about specificity, and I want to now move on to functions. And then the natural question is, okay, so you have these phage communities, they seem to be specific. What are they doing in there? What is their function? And phages, as you know, have, besides the genes such as tails and fibers and head proteins, they also frequently carry so-called auxiliary genes. Sorry, genes that they pick up and move along as they go from bacteria to bacterium. So here we compared the relative abundance of genes on sponge genomes versus those from seawater, and we looked for genes that were enriched on the sponge viral metagenomes. And indeed, we found a number, metabolism for one, also adhesion domains, cell signaling, but we then got intrigued by the finding that ankyrin repeat domains are and seem to be enriched on those phage genomes. And you may be familiar with ankyrins. These are short repeats that are found frequently in pathogens, intracellular pathogens, but also Legionella, that somehow seem to mediate the interaction of certain pathogenic bacteria with the host cells. And previous work by Thorsten Thomas and his team has also shown a relevance of such ankyrin repeat domains in sponge bacteria interactions. So with that background, we were keen to understand more about those ankyrin repeats and what role it may play in the sponge context. And therefore, Martin went ahead and had taken those domains, and then he expressed them in E. coli, and secondly, also synthesized them. And then he went into a big experiment back then done in the lab of Philip Rosenstiel, that you may also know from the CRC and the SymbNET network. So he had this expressed ankyrin domain, and he then tested this ankyrin domain in E. coli cells, expressing the ankyrin domain, and as a control, he had E. coli that was expressing GFP, just as a control. And then he compared the efficiency of such bacteria to resist phagocytosis. And he had several different infection doses. And he found from this experiment, when E. coli was carrying that ankyrin domain, they were more resistant to phagocytosis than those bacteria carrying GFP. So this was the first evidence that we had that this phage ankyrin would somehow translate into a phenotype in this case, phagocytosis resistance. And on the top right, I should say, is a 2D model of the 3 anchoring domains that we were working with back then, right? So we have this phage protein expressed in E. coli, and it seems to confer the phenotype of phagocytosis resistant. And then he did a second experiment. He went one step further and tested in murine macrophage cell lines the host response to anchoring exposure. And he found that certain inflammatory markers, such as TNF-alpha and others, were reduced. So we learned 2 things, and because it's a rather complicated story, I would like to summarize our findings as here, that the anchiphages, anchoring domains carried by phages, are essentially modulators of microbe-host interactions. Modulators in the sense that if a bacterial symbiont carries such an anchiphage that we believe, based on the genomic signatures, is excreted, then it would result in a reduced phagocytosis, and it would result in a reduced pro-inflammatory response. However, if a bacterium was naive and would not carry the anchiphage, it would normally be digested by phagocytosis, right? And if you remember my introduction, phagocytosis inside such sponges plays a huge role, because that's all these sponge cells do. They patrol and they eat what they can find. And we believe now that the phage basically protects this bacterium against digestion by the animal. And that story is a little bit special, because normally phages and viruses are known as toxic and lytic and destroying things. And the normal story goes, kill the winner, and phages are normally meant as lytic players. But here we have a phage that seems to be a modulator rather than a killer. A modulator because it does play with the microbe-host interaction. And if you think about it in evolutionary terms, the phage really has no interest to be lytic, because it wants to live inside the bacterium, and the bacterium wants to live inside the sponge because that's stable and nutrient rich. Then I cannot see you on my screen, but I think you're thinking, why has she not done it in sponges? Why did she go to human cells? And since this question will come, I'm quite sure, just to explain to you that our techniques in sponges are not ready. We do not have primary sponge cells. We don't have immortal cell lines. And therefore, we had to go into this human cell line model. Right. I have one more step to tell you. So that's nice, right? We have sponges, we have bacteria, we have phages, we have certain anchoring domains that seem to have a function. But is it relevant? And then this is now my last data slide. We then queried, and when I say we, I mean, of course, Martin. Martin queried and basically asked, so are we the only ones with anchiphages, or are there more in the databases? And we were quite intrigued to find that these anchorings, the anchoring domains, our specific ones, are quite widespread. And especially here, when you plot by phage environment, you see that the oral, our mouth, but also the gut, the stomach, and other host-associated microbiomes have a pronounced presence of such anch domains. Yeah. So we would like to postulate, and also if you look at the bacteria, Streptomyces, Prevotella, Akkermansia, these are bacteria known from the human gut. So we would like to propose and say that we needed to go into the water and look for sponges to learn about something that may be widely distributed and widely functional also in host-microbe interactions, including our own human microbiome. So what have we learned? We have learned that sponges are reservoirs of novel viral diversity. We have learned a striking pattern in the sense that each sponge has its own viral. It's remarkable because these sponges were collected on the size of a classroom, to say, so there wasn't much distance between them. We developed this phage fish correlated electron microscopy approach. For reasons of time, I did not go into this today, but refer you again to Martin's ISME paper of last year, allowing us to visualize single pages within host contexts. And then in terms of function, we learned that phages encode anchoring domains that we propose help bacteria in evading eukaryotic immune systems. And finally, we believe that such mechanisms, modulatory mechanisms of functions, of phage functions, are much more widespread, including our own human ecosystem. I want to acknowledge my team here at GEOMAR. It's Martin Jahn, who has left, but he's on the picture exactly down here, but he's, as I mentioned, left the lab. I have wonderful collaborators. I want to point out Rodrigo Costa and Tina Keller-Costa, who are in Lisbon, in Portugal. We have many years of working together. And here Peter Schupp, Jasper de Foy, Laura Steindler, and many more. And I want to close by saying that this work was done in the context of the meta-organism CRC, an environment that I enjoy very much working in. And I want to not send you home necessarily, but finish my talk by saying that these marine sponges are just hotspots of diversity and function, and that we're looking at the levels from viruses to microbes to ecosystems. Thank you very much. That's it.
Okay, thank you very much. Are there any questions here? Yeah.
Hi, I have, you may have mentioned it and I missed it, but how do these anchoring proteins interfere with the bacteria in a way that they get less phagicide?
Yes, so the how is always an important question, but also a tough one, right? So we don't know how, but what we do know that E. coli expressing anchorins is less well phagocytosed. And from other studies in human pathogens, we know that the anchorins may interfere with the phagosome-lysosome fusion. So I imagine that the anchophages would somehow interrupt the cellular processes of phagocytosis. And we also know from the second data set that the anchorins reduce the inflammatory response. So somehow, and that's the how part that I cannot answer, they are dampening an inflammatory response. And it would now take a second, third, or 100th effort to understand precisely how they immune modulate. I personally think there is also some potential from the medical field, because the substances that downregulate inflammatory responses, I imagine, is a big medical interest, yeah, in certain operations and pathogenic outcomes.
My question was rather, what do you think it's really on the recognition of the bacteria, or once you have recognition, whether you have modulation of the phagocyte response?
The second, absolutely the second.
Thank you.
Mm-hmm.
I think we have time for two more questions. One is here, and the other is on online by Jonathan afterwards.
Hi. Very nice talk. Maybe I missed it, but these phages are lytic or temperate phages?
In that ISME paper that I didn't go into, it appears that they are largely lysogenic, dominantly lysogenic. So we see very little lytic processes inside the sponge. And that fits to that in this modulatory lifestyle, right?
Okay, and do you know what is the induction signal? Because you have to have the phages outside and form active phage particles with the proteins in the capsule.
The proteins may be excreted without the bacteria needing to lyse.
Okay, nice.
Mm-hmm.
Okay, thanks. So Jonathan, you want to ask a question, please?
Yeah, yeah. Can you hear me?
Yes, very well.
Can you hear me?
Mhm.
Okay. Yeah, thank you. It was fascinating and very interesting. I also want to follow up a little bit on Vera's question about the nature of the inhibition. I mean, there is an increasingly well-known pathway whereby eukaryotic cells prevent themselves from being phagocytosed. They're called the "don't eat me" pathway. And it's elicited by contact with a membrane protein called CD47. It's become a new avenue in cancer therapy because tumor cells express CD47 and thereby avoid being phagocytosed after immunity. And so I just wondered whether it would be worth your while to look into the structures and possible interactions of the ankyrin phage material with mammalian proteins. I mean, it's quite extraordinary that it works in a mouse macrophage and suggests that it's working on a fairly general system. I mean, you said you think it's not phagocytosis itself, but rather the consequences of phagocytosis. But I would like to just see phagocytosis itself having been excluded experimentally.
Right. So in that experimental.
Okay, I can't hear, unfortunately. I can't hear what.
Is everybody else hearing me? I hear myself and I see other people. So I will reply nonetheless, even though. So in that particular study, we did not do any more TEM. So that, and the student has left since then, so we had no chance to go back and do more follow-up experiments. But I think it's well worth, and there's a whole suite of questions one should ask, also in this context of cancer and alike. Yeah, but that project was, you know how it goes. Students finish, they move on, and then came Corona, I should say, and things were different. But,
yeah, thank you very much.
Okay, thank you very much, Ute. I think we're gonna move to the next speaker now. Thanks.
Thank you, everybody. Bye-bye.
Bye.